So I think Alex can start.
Okay, all right. Okay, let me stop the music first. Hi, a very good morning to the Honorable Chair, speakers, and guests. My name is Alex Pang from UMC Medical Center Medical Research Ethics Committee. Welcome to join our webinar this morning for the very special topic today, ethical and regulatory aspects in stem cell research and therapy. Before we start, I would like to inform you that this session will be recorded for the educational purposes. And we also would like to thank for the advanced questions put forward earlier during the registration. Nevertheless, we are welcome your questions uh, during the session later. Please feel free to drop the questions under Q&A box, and we shall do our very best to answer you during the panel discussion. So let me know if you have any further questions. All right, so without further ado, I would like to introduce the chair of the webinar today, distinguished Professor Dato Dr. Lui Lai Ming, the chair of UMMC MREC, to give her welcome speech. Over to you, Prof. Lui. Thank you.
Thank you, Alex. Well, ladies and gentlemen, welcome to this seminar on the ethical and regulatory aspects in stem cell research and therapy. In today's climate, I'm sure that most of us have been inundated with webinars and seminars and crisis meetings related to the COVID pandemic, to ivermectin, to conspiracy theories, and so on. So I expected that there would be a sense of webinar fatigue by now. But I'm truly amazed to find that no less than 250 or more have registered for this seminar. I know that they'll be coming in because right now I think about 100 are already here. I would like to think of this seminar as a seminar of hope and a reflection of the resilience among our community. That in the midst of these dark times, these challenging times, we are still pressing ahead with research, with discoveries and innovations at the frontiers of medical science. So we dare to dream, we dare to hope, and we still do that. Stem cell research and therapy is no longer a matter of science fiction and speculation, and stem cell researchers are making great strides in understanding the normal development of the body, what goes wrong in disease, how stem cells work in the body, and the capacity of stem cells for healing and harnessing the unique properties of stem cells to help patients. And we know that stem cell therapies have been explored to treat a multitude of disorders, ranging from autoimmune inflammatory disorders to neurological and orthopedic conditions, traumatic injuries, uh, and Parkinson's disease, strokes, and so on. But because the public can be easily impressed by new therapies and miracle cures, there have also been many misinformation, exploitations, and abuses in this field. So the crux of the matter is how we, as a scientific community, regulate ourselves to ensure responsible science and the safe and ethical practices in this very promising field. So this is a fairly short seminar, it's only 2 hours, but it's designed to focus on the ethical and regulatory aspects in the Malaysian setting. And we hope will be relevant to researchers as well as those of us who evaluate research. We are very privileged to have very experienced speakers and panelists who will share their practical experience in these aspects. And I will introduce each one of them as we go. So instead of introducing all of them at once, I will sort of, just to break the monotony, I introduce them when it's time for them to speak. So in this seminar, we will not be delving into the deep science of stem cell research and therapies. If we do that, it'll probably take a few days. But it is important to have, nevertheless, an idea of how far we have come in this field. So to start that off, we are privileged to have Professor Tengku Kamarul Zaman to provide us with an overview, and he will start off the seminar as the first speaker on the topic of current stem cell research and therapy landscape in Malaysia. Let me just say a few words of introduction on Professor Dr. Tengku Kamarul Zaman. I think many of us already know him, but it is still proper and good to introduce him. He's the former director of the University of Malaya Medical Center, and he's the current director of Institute Perubatan dan Pergigian Termaju in USM. Besides his training as an orthopedic surgeon, he holds a diploma in tissue banking from the National University of Singapore and a PhD from the University of Liverpool. So he's a unique person. His research interests include tissue banking, transplantation, tissue engineering, and mesenchymal stem cell applications in orthopedic surgery, with a practice focus on regenerative medicine. He's a strong advocate for orthopedic research and is chair of the Asia Pacific Orthopedic Research Society and vice president of the ASEAN Orthopedic Research Society. He's a fellow of the Academy of Sciences Malaysia and serves on the National Committee on Ethics of Stem Cell Research and Therapy. So, Prof. Tengku Kamarul, the floor is yours.
Thank you very much, Prof. Louis. Thank you for your kind words, Prof. Louis, and a very good introduction. I won't take too much time here, so if you can, Alex, I need permission to share my screen.
Yeah, I think you can share it now. Yeah.
Host is available, participants screen sharing. So
Okay. Yeah, sure.
I still can't.
Can you? Yeah.
Okay, very good. Okay, thank you. Okay, just give me a minute. All right. Are you able to see the whole screen?
Yeah, we can see the screen now.
Okay, very good.
Yep.
Okay.
Thanks.
So let me begin. So I've given this task to talk about current stem cell research and therapy landscape in Malaysia. And I think Prof. Louis has made it very clear that we're not going to delve into the science of it because it will take the whole day, maybe not even a whole day, probably the whole month if we continue to talk about it. So I'm just going to touch here and there just to give a brief idea what stem cell research and therapy is like in Malaysia, but giving a whole global view of where we are today. Sorry.
Yeah. Hi, Prof. Maybe you can put the full screen there, the slideshow. Yeah.
Oh,
if
sorry.
you want to.
Okay. All right.
Yeah. Thank you.
Give me a minute. Is all this the problem here? Give me a minute. Okay, just give me a minute.
Sure.
Not to worry, we're actually a little bit ahead of time, so you're all right.
Let me just put this first into full slideshow. Okay. And then let me just share this screen. Okay.
Yeah, you share the screen first, then you enlarge with the slide. That's great. Thank you.
Okay, right. Okay, very good. Thank you. Sorry about that. Okay. So I'm just going to talk a brief global overview of the topic so that you can understand where we are today and try to move home into the context of where Malaysia is today. All right. You see, okay. So, for whatever reason, it's misbehaving. Sorry, just a minute. Okay, hold on. I'm not sure why it's misbehaving. Sorry, let me just try this, okay? It's just misbehaving, sorry. I'm not sure why. Hold on. Just give me a minute, yeah, hold on.
No worries, no worries.
It seems to work here. Hold on. Let me just try this. Okay, are you seeing the full screen? Yes?
Yes.
Okay, let me just try this again. Let's see whether this. Let's see. Yeah, okay. So let me just begin. Okay, sorry about that. So what are stem cells? So stem cells are cells that from which all of the cells are specialized and they are generated from. And what's to say is these cells are not differentiated. They come, they are having ability to go into many lineages, and they are like raw materials. This is not my own words. I've taken this from the Mayo Clinic actually, and they call it the raw materials that they allow the cells that go into the sites and results in tissue repair. And hence, in many studies and many centers, they see the stem cells as a source of tissue repair. And the interest is because now you have the power to harness cells to provide a source that you can now repair all the damaged tissue, and as you grow age, you can replace the aging tissue. These are what stem cells hold promise. And basic characteristics, just two things actually. Stem cells can undergo self-renewal, means they can just continuously grow and renew themselves to form new cells that have the same function. And they have the ability to differentiate into the type of cells that you want it to repair. So the idea is if I had a muscle tear, I can put the stem cells there and the muscle will just grow back. That's the general idea of what people have. And how many types of stem cells? Well, generally, if you listen to every talk and look for every website, they talk many types of stem cells. But literally, the one that you have to talk about is actually either adult or embryonic stem cells. So if you look into the picture to the lower right box I put down here, so if you look at this stage, any cells above the embryonal layer above is considered as embryonic stem cells, and once you're born, anywhere beyond that is called adult stem cells. Now, when you bring it out to adult stem cells, there are two things that may come to mind. It's either you talk about hematopoietic or mesenchymal stem cells. Most commonly seen in most studies where they say we've got a stem cell center in the hospitals, especially, they're talking about the hematopoietic stem cell center, which they do for blood transplants, bone marrow transplants, etc. So things like leukemia, thalassemia, all of those, aplastic anemia, those are now well-established therapies. So the one that really comes into mind for most people when they talk about stem cell is those non-hematopoietic stem cells, which is either the adult or embryonic. And in most recent years, well, not more recent, but 10 years now, they're now talking about adult stem cells that can behave like embryonic stem cells. And what you're talking about these stem cells is that they can be programmed to behave like embryonic, but they're actually from adult stem cells. So just to give you a bit of overview, I'm not going to go too much to the science. The embryonic stem cells, you need to get a whole egg, a whole organism to let it fertilize. And at the stage within day 6 or day 7, you have something called the inner cell mass. So you actually kill or destroy this zygote in this early stage to take out the inner cell mass to do for therapy, as opposed to adult stem cells. If you're embryonically formed, if you talk about the trilaminar layer of the embryology stage, which is ectodome, mesodome, endodome, any cells that come from the mesodome is considered as mesenchymal stem cells. And because most of the body, 90% of our body comes from the mesodermal layer, and therefore, theoretically, I can take some of the bone marrow or fat and try to extract these stem cells. So adult stem cells appears to be a lot more acceptable to many because it doesn't require any destruction of any live cells. Okay, so that's the reason why there is an ethical issue when you talk about embryonic stem cells, and hence to overcome, because you want the potency to be high, like this, as you talk about high potency, it's like pluripotency, which you find in embryonic stem cells. You want to have that level of potency in a safer sort of cell without the ethical issues. So that's why they come up with the iPSC, right? So why then you ask is the focus is now talking about mesenchymal stem cells, And that's the reason for the rest of the talk. I'm just going to talk about mesenchymal stem cell because I think when they talk about stem cell ethical issues and where the landscape is going in Malaysia, they're not talking more of the mesenchymal stem cells, although a lot of people talk about umbilical cord and all that. They actually fall under the adult stem cells, and they literally have the same characteristics of mesenchymal stem cells, and that's the reason why it becomes a focus. It's a lot safer, it's easier to extract. It doesn't have the problems that you come with embryonic stem cells and the ethical issues that comes. So there's a lot of interest to this. But always, there's always an issue about age-related problems. They've got the use of it being used for non-homologous use because, for example, if I were to take from bone marrow, I don't put it back in bone marrow. I want to use the bone marrow stem cells to go and repair my cartilage, my tendon, my bones. That's the non-homologous use. And of course, these supplies also age as you grow older, and they've got limited resources that you have to do. And most of the time, these stem cells require them to be grown in a lab. So these are some of the issues that comes with mesenchymal stem cells. But despite all of that, it's still being the ones being preferred by most industries. So I've got the nice slide here that goes for about a minute, trying to give an overview of what stem cells all about. And it's actually a very nice slide talking about the stem cells that can go under self-renewing and differentiation potential and can form all these tissues that you see here. And in a nutshell, as I said before, you break it down and say that there are either the type of stem cells, which is embryonic stem cells, which come from the inner cell mass, which is very potent. You have the somatic cells, and what you can do is you can get the multi-scopal stem cells that does the function of that. So multipotent, as I mentioned before, there's hematopoietic and mesenchymal stem cells, and the hematopoietic stem cells literally is meant for just the blood disorders. So the MSCs come into many actions, and there is many ways that they talk about the actions of the stem cells, how they repair, either through the immunomodulatory activities as shown in these pictures, or they have something what they call close or cell-to-cell contact signaling. So it means the cells goes near to another cells, they then mediate the cells to undergo repair. On the slide, this is another image as you can see here. This is how the cells can grow, and the cells can grow pretty rapidly. In our lab, we can grow in 6 weeks, we can get millions, about 60 times the number you have cells, and you see pictures from there. They can sprout and form all kinds of cell shapes. And they have a criteria which is well established to tell you that these are stem cells. I won't go into details, but this has already been proven by the International Society of Cell Therapy. Now, what is the global potential market? I've taken this from Model Intelligence, and they said that the stem cell market today is valued as 14.7 billion. And about 30% of it is actually hematopoietic. In fact, they said that a bigger proportion of it is now going into the mesenchymal stem cell or adult stem cell market, which now covers almost 70%. So they said that the annual growth rate or CAGR can come to about 10.24%. That's even higher than most of the inflation rates in many developing countries. Okay, so and I said just now, while the oncology segment is being covered by the hematology, but the other segment that we're talking about today would be talking about the non-homologous or adult stem cells, right? Now, I did a very quick search over here and did a survey on the PubMeds for Malaysia. And looking at this, I had to go through about 1,161 results, and they're correct. They're all from Malaysia. They have at least one author there. I would say that some of it, I'm looking at it, probably the authors actually just put their names there. They're not really being the work being done in Malaysia, but I assume that's still Malaysian anyway. So we take it as face value that 1,651 results. I broke it down. And really, interestingly, it's actually to show that in terms of clinical trials, and it's only 15 of them, all right, and 15 of them. And most of the studies are either, a lot of it is in vitro research, which quite a number of it I've contributed myself in this. There are reviews, systematic reviews, quite a huge number of it, but clinical trials and applications only about 15 being published. When I broke it down and look into this, almost actual numbers were actually 13, because there were journals who kept publishing the same results twice. For example, I did 20 patients, I published one time, and I did another journal, the same journal published, oh, now I've got 40 patients, now I'm going to publish it a second time. So I've removed those two, okay, and published twice, then actual number is 13. Only about 7 of them actually on hematopoietic stem cells. So I said, these are well established in combination with chemotherapy, immunotherapy. So the one that talk about adult stem cells are all of these, okay, there's about 6 of them, right, that talks about vaccine studies, SLE, hemophilia, ischemic limb, and cartilage, and all of this is published in Malaysia. right? If I were to break down and look into some of the studies, this is from the clinicaltrials.gov website, and you can see that the MSc market is actually quite big, and quite a number of them are looking at a number of disease, as mentioned by Prof. Louis, looking at clinical trials. They are registered but not published, right? And I've got this good paper by Aina Kaplan. It's a very interesting epidemiological paper that looks into all the trials and published in 2020, August, about a year ago. So I assume that this is valid by 2019, right? Okay, because it takes time to get things published. And you can see the clinical trials in MSc studies are actually quite high, and it's on the rise, right? And if you look into this, the highest number is traumatology. When I looked into the paper, the traumatology actually refers to my field, which is orthopedics. So 234 trials are doing this. And quite a number of these studies in worldwide, we're talking about registered worldwide, is in phase 2 and phase 1 actually, all right? And they're still quite far from reaching phase 3. Malaysia, interestingly, this paper is published here, and I think it corresponds quite well with the registered trials and the publications that we've seen so far. There's 8. I tried to break down, they didn't give the exact details of the data, but I assume that Malaysia is also in the game of clinical trials as well. So these are some of the examples of what has been treated, the kind of sources. As I said, they're mainly all MSCs, and people talk about umbilical cord, adipose derived, they're all MSCs actually, right? So it's just where you extract them and how you use them, right? Okay, I won't go into details. As I mentioned before, the studies looked into mesenchymal stem cells to form other soft tissues. Osteocytes are for bone, chondrocytes for cartilage, pancreatic cells is for pancreas, and hepatocytes for liver. So they've used for a number of diseases. But again, as I said before, quite a number of the studies are now moving towards myocardial infarction as the way forward. In fact, there are quite a number of clinical trials published on myocardial infarction, right? Now, looking at this race, a number of publications and whatnot, I'm looking at the drivers that will determine the research in MSc in Malaysia, and be very, very clear that it is a market demand sort of study. So literally, while certain studies and applications may show efficacy, it may not be the best thing that you can think of, because a lot of times as a research or something being done is market driven. And there's a lot of studies nowadays, they talk about using adipose derived stem cells where we can prove that bone marrow may be better. But because adipose, in especially Malaysians where 50% is either fat or obese, it's so much easier to go and extract bone marrow from adipose than it is from bone marrow. And this is becoming a more a lucrative business to undergo. And that's why we can see quite a number of studies are moving towards that, because it's very market driven as today. So there are also controversies in MSc, and as Prof. Louis pointed out just now, this is something that we really have to address. And in fact, some of them mask themselves as being real science, okay? What I call as real pseudoscience or pseudo-real science. We're not really sure, because sometimes they use nice jargons, and if you think about it, this sounds logical, but when you look into the science behind it, it doesn't really make sense. And I've looked into the studies talking about MSCs in COVID-19, because they're saying that we can do the immunomodulatory effect. But the direct effect of it, of the study, I've not seen the basic science behind it, and it's a lot of hearsay. So while there's quite a number of these needs to be proven, there's a lot of things that needs to be discussed and looked into with these MSCs. And that's the reason why we also have to differentiate, at least that has some scientific basis to it. And these are available on Malaysian websites. You can see the word Malaysia up there, and reverse aging process. And these are being offered to a number of clinics here in Malaysia. It's not being well regulated. I'm not saying that a lot of things are bad out there, but a lot of things like this are not really proven. And in fact, you ask me between this and that of the studies talking about MSCs in COVID, I would prefer the MSCs COVID. At least that makes some sense. None of this actually makes sense, actually. Okay, they're just harping on the name. And that's the reason why in Malaysia, today's landscape, we talk about having to have a proper regulatory control of the current conditions and having it under the authoritative body. I won't go into detail. I understand there's a person from MPRA who's an expert who will talk about this. But having said this, I'm just going to touch base and tell you that basically there are things like guidelines on stem cells. There are things on research services. These are available from the websites. You can get it from the National Library. And I think anybody who wants to embark in stem cell research and therapy should look into this. In fact, it's also been published in the Malaysian Medical Council, which they also hold a copy of this, of their own guidelines of how doctors conduct research and therapy. In Malaysia, under Professor Datuk Rahman Jamal, he is the chair for the NSCERT. Now, I'm not really sure because I'm part of the committee. I think it's now called NSCERT, but on the website it's called NSCERT. So it's not really stem cell anymore. It now covers a degree of cell therapeutics and advanced therapies. I think I'll let Datuk Rahman Jamal try to explain that. But the idea is so that if there is research, there's something to be done. There's somebody who oversees this and overviews the ethical use of stem cells. Ethics not just for the issue of the embryonic stem cells, as I mentioned before, because you're going to kill another cell just to get the cells for your therapy, but rather you need to talk about as the issue that I said, whether there's any proof to having paid so much to do a treatment that has not shown any efficacy or very low efficacy. Is it ethical for us to do? So somebody has to oversee this and make sure that there is standards in such applications. These guidelines even was announced many years ago. I was involved with the MPRA since 2016, and this was published in 2018 that this year that is going to be applied. I think it's going to be applied. Now it's now a stage of voluntary registration. I'm just going to touch it a bit because somebody else is going to talk about it. There will be 4 divisions that look into this. There will be the medical development divisions, a medical practice division. They will look in the clinical use. There will be the medical device authority, which I remember having to discuss this between cell therapy and tissue engineering. Where does it go? So if it's tissue engineering, it's usually a combination of cell and the material themselves, the device. So they said that if the main function is on the device, it falls under the MDA. If the main function is on the cells, it falls under the NPRA. All right. So that's the idea is. In all these documents, the big thing they talk about is class 1 and class 2, and meaning to say that class 1 is manipulated. If you talk about class 2, anything else not class 1 falls into class 2, and that literally puts 99% of all the cells in the market that are being used today falls under class 2. So I won't go into detail of this detail, and if you go into class 2, there's a lot of things that you need to really fulfill the documentation and dossiers in order for you to pass through a regulatory process. Just in a nutshell, this was given to us by the NPRA. You need to have to comply with the class 2 classification, which you need to prepare the full dossier. Again, I will leave NPRA to explain what is a full dossier in this case. And what happens if somebody ignores these rules and regulation? Well, as I mentioned before, there will be the medical practitioners, Bahagian Amalan, Ministry of Health. Last time was Dr. Razid. Today is, I think, is Dr. Azman. They will have to go and chase the doctors for having to transplant, not fulfilling the pharmaceutical bid. The Malaysian Medical Council has its own guidelines, and therefore, if there's a complaint, they need to go and ensure that the doctors answers for them. I remember there was a case where there was stem cell injected into a lady's breast, and the doctor was going to see was one German doctor and another local fellow. The German doctor just left the country because he was going to be charged, and I think the local doctor, I'm not sure what happened to the case. That was many years ago. Something went wrong. So the medical council comes in. And of course, for manufacturers and laboratory operators, there's the Pharmaceutical Act, the Sales and Drug Act, that all is governed under the new CGTP that's been proposed. There's the CDCR 1984, which is the drug and cosmetic usage. And of course, there's the MDA or Act 737. The issue is with all of this, and we've discussed this many times, myself, Datuk Rahman Jamal, and a few other players, who's going to enforce all of this? And Malaysians well known for having so many regulations and acts, but when it comes to enforcement, there'll be a very gray line that is, oh, this is not under me, this is not under you. So quite a number of times the enforcement is not being taken out very seriously, all right, in Malaysia. Now, regulation for me, I personally feel is important. Although people say that we'll stifle the development, I said, look, we allow development to occur. We're not stopping anybody back, but somewhere along the line, it needs to be regulated because there must be a view that the safety for the rakyat is being taken care of. We need to be sure as a national level, as Malaysians, we need to uphold certain ethical and moral values. That gives a very good international reputation for all of us. And if you're talking about having to support sustainable growth of this market segment, it is very important that these kind of views are being seen very favorably by the community worldwide. Okay, so for me, regulation is quite important, but not to stifle the development, but rather to support it and make it a more reputable business. Okay, I've did my bit. I've gone around having to go and even appear in Chinese newspaper in Astro Awani, trying to say people that you need to go and do this. Somehow I will never beat these two beautiful ladies, as you can see here. I'm not really sure what makes them qualified to talk about stem cells, but people will believe that stem cells I'm talking about comes from trees and apples and God knows what. Okay, they are not being regulated anyway. So in Malaysia, these sort of things still happen. So I even given up having to tell people about it, because the public really needs to see a pretty face in front to go and talk about it. So maybe we have to find somebody who's a professor who looks very pretty and very strong public opinion can pull over to go and explain to them what stem cells all about. All right. So what awaits in the future? I'll be very quick. I'm sorry, I've taken few more minutes here. What awaits in the future and whether it's going to be regulated? We can see in the near future that there are going to be not just stem cells being taken, transplanted into sites to cause repair. They will be manipulated in many ways. And this is something that the MPRA will have to be sure and understand whether or not it falls under this, or they will decide to say that this falls under a different category of drugs called biologics. If it's a biologics, then it become a different regulatory pathway altogether. Now, one of the things that we talk about, and I've seen quite a number of studies, is talk about the exosome derived from MSCs. They are not really the cells, but rather the content. I was told that any exosomes is now falls under biologics, under this is going to be a drug. Whether or not it's going to be a true drug or not, this is something that needs to be debated. But I can see quite a number of studies are doing this exosomes. In fact, in my lab, we're now taking exosomes, but not from MSCs, but rather from platelets as a way of trying to use to harness the potential of the proteins inside the cells. The next thing is whether or not allergenic sources using umbilical cord will be the biggest vaccine, because now if you look into certain studies or certain websites or certain papers, they talk about rather than adult and embryonic stem cells, they talk about something called perinatal stem cells, and they say that this is the best. For me, they are still following the MSC. They're actually just MSCs. It's just that between the MSCs, these are young MSCs compared to somebody who's taken the MSCs from my body, which is probably old, and they have higher efficacy. The question now, this is something that the ethics committee has to ask whether it is ethical enough to go and take this at the stage where it is the crossover between embryonic as well as human. So this is something that will be seen in the near future for the use of MSCs. Then there'll be the alternative source. Our lab have been talking about peripheral blood derived. Quite a number of people now claim that they have done the peripheral blood. Take it from me, it takes a lot of effort trying to get MSCs from blood where the population is less than 0.1%. To isolate them. And what they've done is most studies actually talk about hematopoietic stem cells to pass them off as hematopoietic stem cells, and something needs to be regulated because it falls under the hematological stem cells, then it no longer falls under the CGTP. This is something that we'll look into in the near future. The next one is genetic manipulation. So CGTP talks about it, but didn't talk about how about cells that have undergone some sort of targeted gene editing rather than going into gene therapy itself. So this would be the near forward, because CRISPR technology is already here, and it won't be long before you can see cells being manipulated using CRISPR. And last but not least, we talk about induced pluripotent stem cells that's being used. And although mainly induced pluripotent stem cells are being used just to do for screening purposes and whatnot, it is now being seen to be used for conditions which is now talking about salvage surgeries and compassionate use. So whether or not these are going to be regulated, we have not very clear if it falls under the first category, 1 or 2. I'm just going to mention a bit here, but I'm not really too sure that we'll see in this future. We're talking about specific targeted pathways. Now, the stem cells, what they do is they target the specific pathways of the genes themselves, either by blocking them using chemicals or anti-proteins that targets intracellular molecules. But this will be something that you have to understand as well. It's going to be manipulated in a certain way. And while you say it's class 2, you cannot really say that they are class 2 because they're literally just going to be cultured in a very simple manner, all right? Or they can even be treated in the patient before they've been extracted. So does this fall into type 2? I'm not sure. So this is something that needs to be debated further. So in conclusion, right, okay, stem cells is a very growing area, all right, and especially for the non-hematological stem cells, as I mentioned before, because HSC is being used by all the centers around the world, so there's nothing really new about it. But MSCs will remain a big focus because of the safety records, although the embryoning and iPSC is also coming strong. Certain areas, as I showed before, they're moving to phase 2 and phase 3 very rapidly, and there's going to be an emergence of all these drugs in the near future. And this is something that Malaysia will probably have to deal with in the next few years. But if you're talking about some of you in wanting to go into the market-driven process, your research and your work will need to go into a streamline towards the market-driven processes. And hopefully that if you do that, then it will be more accepted by the industry. After all, money talks, you know. So all these things that you have has to be translated to make it work. I'd like to thank the organizers of this event to invite me to give this lecture. And I'm quite happy to go and discuss this further. Thank you very much.
Thank you, Prof. Tengku Kamaro, for that really amazing and exciting overview. You've covered so much field actually in this brief time. I'm just amazed at how you managed to do this. But basically, what you have shown is that how researchers can dare to dream of so many things, bring them to reality, but you've also emphasized the need for regulation, which is extremely important. And I'd just like to let the participants know that we won't take questions at the end of each talk so that there is time for everybody to speak, but we'll throw all the questions to the last part of the seminar, that's at the panel discussion, so that different speakers also can give their input to the questions that are raised. So I'll just move on to the second talk straight away. And the speaker for the second talk is Professor Dr. Kamal Jamal. He's a professor of pediatric hemato-oncology and molecular biology at UKM. He has his PhD in hematology and molecular biology from UCL, and he's a key driver of molecular medicine research in Malaysia. He's the co-founder of, he's the founding editor of UMBI and has helmed its initiative since its establishment in 2003. So he's an iconic figure whom I think most of us know. So besides being an accomplished researcher, he sits on numerous committees, and Prof. Tengku has mentioned that Prof. Raman Jama is the chair of the National Committee on Ethics of Stem Cell Research and Therapy, but he also sits on the National Committee for Thalassemia Registry in the Ministry of Health and the National Committee for Clinical Research, sets the policy and directions for clinical research. He's also a fellow of the Academy of Sciences, Malaysia, and he chairs ASM's task force on precision medicine. Prof. Raman Jama is involved in another meeting right now, but he has sent his pre-recorded talk, and we're pleased that he can join us for the panel discussion later. So Alex, can you load his talk on the topic of ethical issues in stem cell research from the perspective of the IRB and NSERT.
A very good morning to all the participants. Thank you to the organizing committee for inviting me to give this talk titled Ethical Issues in Stem Cell Research, Perspectives from the IRB, IEC, and also the National Committee and CERT at the Ministry of Health level. Well, for stem cell research and therapy, which is one of the most exciting innovation that has appeared in the past a decade or so, I believe it's about striking a balance. On one tip of the balance is we are looking at the increasing consumer demands, especially for unmet medical needs or the lack of treatment for some conditions. The promising innovation that is involved in stem cell technology, and also to fulfill an unmet medical needs. And on the other tip, on the other side of the balance is the need to regulate and of course looking at the ethical issues in stem cell research and therapies, issues of safety and efficacy, especially evidence that would support it, and of course, providing the public the protection that is needed when the clinical therapy is approved. Well, this is a typical proposal for approval. I've chosen the title mesenchymal stem cell for the treatment of osteoarthritis, one of the most popular indication now for research. And I believe any of us who sits in the ethics committee or even as a medical doctor, we would ask these questions. Have preclinical studies being done? Do we know at least some of, or at least the mechanism of action or actions? And the next question would be, what would be the dose of cells to be given, the route of administration, whether the tracking of cells have been done, and then purity of cells, and etc. Also, the issue of the use of comparator arm, if this is randomized control trials, and also the need to look at the proper study endpoints and long-term complications, including what many of us worry about when we're using stem cell, which is cancer. The philosophy and approach of guidelines related to stem cell has practiced by most IRBs, IECs, and also regulatory committees is about the promotion of an ecosystem that would focus on the ethical, practical, appropriate, and sustainable enterprise for stem cell research, so that the development of cell therapies should not be inhibited. Hence, the second bullet, it should not be inhibited to innovative therapies and also to fulfill unmet medical needs. And also, it should complement existing legal frameworks and acts. Integrity of the research initiative. This is mainly the responsibility of the investigator or the team of researchers. One, it should be conducted by qualified investigators using rigorous scientific approaches. Number two, ensure that information obtained will be trustworthy and reliable. And it is the duty of the investigators to submit for an independent peer review and oversight, and not just to go on with doing the experiment. And last, ensure that primary goals are clear and beneficial, and that they address unmet medical needs, which I have mentioned a few times already so far. I just want to highlight briefly about human embryo and stem cell related research in relation to embryos and embryonic stem cell lines. These are rare in terms of submitting to our own local committee, but I just want to highlight some of the points from the latest International Society for Stem Cell Research, which is published in 2021. I would like to encourage the participants to download and read this. And there are approval or applications that are allowed as before, but there are also emerging applications that have been sort of listed in these guidelines. The experiment on in vitro use of pluripotent stem cell and also in vitro organoid research is allowable, as most countries would agree on this, or most regulatory committees would agree. And the need to review by a special oversight committee. This would include procurement of embryos for in vitro research. This has not changed much, as well as the derivation of cell lines from embryos. Those that are still not allowed is heritable genome editing. This has been discussed in most countries, and there are international guidelines for this. WHO and also the National Academy of Sciences of USA has also published this guideline online. And also human reproductive cloning. This is still not allowed. So let's go into the therapeutic use of stem cells. So this is, of course, the most exciting area now and most popular among the protocols that are being submitted to the IRB and IEC, as well as the answer at the Ministry of Health level. There are two key points here. One is making the approval for clinical trials, and number two, looking at the aspect of safety and efficacy. So any committee looking at this proposal should only approve the proposal for clinical trials only when there is a compelling scientific rationale and a plausible mechanism of action, and an acceptable chance of success. For the safety and efficacy part, this must be demonstrated in well-designed clinical trials with approval by regulators before the interventions are offered to patients. So in the use of stem cells, there are three main contentions or three main groups or three main applications that we usually discuss. And these are the areas where the IRB, IEC must be familiar with. Number one, the use of minimally manipulated stem cells or tissues. Number two, the use of substantially manipulated stem cells or tissues. The third one is the use of minimally manipulated stem cells or tissues for non-homologous treatment, which is rather complex and speculative. So these are the three main groups of use, and we should be familiar with this. Definition of the minimally manipulation or minimal manipulation is available in many websites and also at our National Pharmaceutical Regulatory Authority. For the minimally manipulated stem cells or tissues, these are generally exempted from regulatory oversight, but the responsibility rests on the clinician or the investigator to invite independent scrutiny of their process of manipulation. They cannot assume that what they have done is minimally manipulated. It's good practice to invite an independent scrutiny to decide on the level of regulation required. And if it is uncertain or in the presence of disagreement, it is best for the IRB and IEC to refer to the regulatory agencies. And for Malaysia, this should be referred to the NPRA. For substantially manipulated products, there is a need to determine the safety and efficacy profile using the rigorous research methods. This again cannot be assumed because the composition of intervention may differ from the original tissue source because they have been manipulated. Number two, these products are evaluated as drugs, biologics, and advanced therapy medicinal products, the guidelines of which are available at for our local application to the NPRA guidelines. Lastly, the non-homologous use of stem cells and tissues. This clearly has potential benefits, but can also pose serious risks. As many of our researchers know, there has been many incidents on this, where one example is the use of adipose tissue-derived stromal cells for the treatment of macular degeneration. A publication in 2017 showed it has caused, this kind of intervention has caused the loss of vision. Number two, there is a need to show the benefit-risk ratio. This is always what IRB and IEC and also NSERT are looking at for the investigator to show the benefit over the risk. I cannot stress more upon this, the need to show safety and effectiveness through preclinical and clinical studies. And this is another aspect or issue that is always of contention at the committee level. The cell processing and manufacturing part. This is basically for us to look at the integrity of the cell's function and safety. First and foremost, the donor consent and screening must be properly documented, because many companies or many researchers are collecting stem cells and then expanding them into master cell banks. So the donors are important sort of stakeholders here, and proper documentation must be shown in the protocol. For manufacturing, this will be alluded more in the talk later, but I just highlight a few things here: quality control systems, and of course, manufacturing under GMP conditions. There are key tests and assays that should be done, including for mutations, characterization, purity, and so on. The components in the cell culture or preservation that is used, because we need to look at xenogenic material exposed to the recipients, as well as exposure to infectious agents. Last but not least, the criteria for product release. We need to look at the final product testing, validated assays that assess the identity, purity, sterility, activity, and potency of the product. Okay, go on to the preclinical study. Again, this is always one of the weakest link and weakest part of some of the proposals submitted. Let's go through this. So any investigator into this area should show rigorous evidence of product safety and establish the proof of principle for the therapeutic effects. And that's the need to use appropriate in vitro and or animal models. Investigators must provide precise, accurate, and unbiased measures of potential clinical utility and internal validity. The animal model or the model used must be as representative as possible of clinical scenarios that they are intended to model, and they should be able to be replicated. For example, if we are using the stem cell for intervention or treatment of Alzheimer's disease, the mouse model must be one that mimics the Alzheimer's disease in human patients. Last but not least, again, another reminder, there's a need to select animal models that best match the human disease, use outcome measures that best match clinical outcomes, and evidence supporting a mechanism of action. So, but there are also some considerations for IRB and IEC to take into consideration for these animal studies or preclinical studies. One, homologous cells in the same species may be unavailable. So if this is justifiable in that protocol, then we should consider that in our deliberation. Number two, immune suppressed animal models, which is often used, do not permit an understanding of the effect of the immune system on transplanted cells. Hence, the full biological mechanisms may not be able to be provided by the investigator. The third contention or consideration is it is not always possible to extrapolate cell therapies in an animal model to humans. Safety studies. These are key studies to perform and to show in the dossier or in the document for clinical trial approval. Preclinical studies must be done in a GLP approved laboratory. Cell characterization should be done. Studies to look at tumorigenicity also must be done and the data shown. Biodistribution studies, long-term safety studies, as well as the assessments of the delivery procedures. Either it should be directly injected or any surgical procedure to perform for the implantation. Efficacy studies, these are the key requirements or key evidence required. Clinical utility, optimal condition. Dose is so important. Many researchers fail to show why the dosage has been selected. Most of them just quoted a previous publication, which again do not contain the justification for the dose. Co-interventions, and I've mentioned also the delivery approach that will be used. The ability to modify disease in an animal model, and it must be of a sufficient magnitude and sustainable in terms of the duration of disease modification. Last but not least, again, the mechanism of action. At least show some, or ideally, a complete understanding of the biological mechanisms at work. We understand that this may not be possible to show in very serious disease for which there have been some evidence in the animal models to show some mechanism of action in terms of how the stem cell would work to repair the disease or damaged tissues. Clinical research, these are the key requirements that any committee, including ANSET, would look for. First and foremost, of course, the proposal must be reviewed by independent experts who are competent to evaluate. We must look at risk-benefit analysis. Potential benefits to subjects must be estimated. The stem cell intervention itself must be clinically competitive to current therapy or meeting an unmet demand. Thorough systemic appraisal of current evidence must be shown by the investigator. Proper choice of comparators, including the best or current best intervention at the clinical level. The use of placebo is possible, and the use of sham comparator. There is a need to report adverse events. We should not hide this, and investigators should be honest to report this to the IRB, IEC, or to the National Committee. Investigators must also publish the results in full, even if the findings are negative. And last but not least, this has come up, occasionally, patient-sponsored and paid to participate trials. Generally, these are usually not allowed, but there is a need to look at the national regulations for this. Okay, I just want to sort of highlight some examples of the issues and concerns in the research protocol submitted to our national committee. Firstly, the use of the non-homologous use of stem cells. This is always complex and not easy to assess. Number two, the lack or absence of preclinical data and studies, especially from the investigators' own lab and own experience. Poor study design. So this is again something that sort of keep on recurring, and I believe this can be improved with the help of a good biostatistician or somebody who's familiar with research design. The study site not licensed for the procedure. So for this, remember, for the private centers, the Private Facility Healthcare Act comes in under, and this is governed or sort of monitored or approved by our CCAPs under the Ministry of Health. Next is the comparator arm used, which is not a standard treatment, so beware of this. And lastly, in my example, little or no explanation at all on mechanism or evidence of mechanism of action. So in summary, we know the applications of stem cells, is very, in terms of inviting, and it showed a lot of promises, but certainly we need more compelling evidence via properly done clinical trials. Secondly, researchers should adhere to guidelines to ensure approval of the research proposal. Issues of safety, quality, and efficacy must be addressed adequately. And we know guidelines may need to be reviewed and revised according with new knowledge and advances. So these are the references that I've used for this talk. You can refer to this, the Guidelines for Ethics in Stem Cell Research and Therapy, published by our Ministry of Health in 2009. And of course, the latest guidelines from the International Society for Stem Cell Research. With that, I thank you very much for your attention. Thank you.
I think we're very grateful to Prof. Raman Jamal for that very comprehensive talk. Actually has covered quite a lot of areas and perhaps have already answered some of the questions that are being put forward by the participants. Since it's not here in person for me to thank him, I'll thank him later when it comes to the panel discussion. We move on to the next talk, and this is on the regulation of stem cell therapy in Malaysia, and I'm very privileged to have Dr. Zaril Harza from the National Pharmaceutical Regulatory Agency, MPRA. He's a Senior Principal Assistant Director at MPRA and current Head of the Evaluation and Safety of Investigational New Product Section. He has experience in the evaluating investigative products used in clinical trials in Malaysia before the issue of the clinical trial import license and permits for exemption to manufacture for local manufacturers. So experience is very wide. It includes the monitoring process for all SAEs, adverse drug reactions, drug accountability reports, closing study reports, and also experience in audit and inspection of trial sites of sponsor and contract organization facilities to ensure adherence to good criminal practice guidelines and other regulatory requirements. So he comes with a wealth of experience from the regulator side, and it's a privilege to hear him speak to us today on the regulation of stem cell therapy in Malaysia. Thank you.
Assalamualaikum, very good morning, everybody. Can you see my slide? Is it in presentation?
Yes, we can.
Is it a presentation view or full form?
It's got the speaker's preview as well.
Oh, okay. Hold on, yeah. Let me change this. Okay. Okay, can you hear? Can you see these slides?
Yes, we can
Okay,
see that. It's
right.
very good.
Okay, right. So I begin my presentation with of course the title of the content of this presentation. So this will be revolving around the regulatory framework for CGTPs. For this particular case, we'll be discussing about the clinical trials requirement, specifically in terms of what constitute what warranted research that needs to be applying for clinical trial import license and clinical trial exemption with regards to the CGTPs. Okay. Okay, I'm sure that every one of you knows in terms of all these drug developments in drug development process. Okay, comes with the clinical trial up to the point of registration. So where does the MPRA rules or jurisdiction falls? So in all of this presentation, we'll be discussing about, rules and regulation acts that you need to adhere to in order to conduct clinical trials involving CGTPs. So for this particular part, we are here in terms of, if you can see at the lower side of it, at preclinical, clinical trials, and up to the point of you're applying for regulatory review. So this is where we falls under clinical trial import license and clinical trial exemption. Apart from that, you also need to adhere to, as what Prof. Ramajaman already said, good laboratory practice in terms of your preclinical studies, as well as your good clinical practice in terms of your clinical research that you are doing for your investigational medicinal product. For this particular case is CGTPs, yeah. So what are the guidelines are related with clinical trials in Malaysia? This one including CGTPs, because we just recently make it mandatory in January that all CGTPs need to be registered with Malaysia. And when it is registered, it comes as well with the requirement for conducting clinical trials when they need to apply for CTR and CTX. So we have three guidelines here. Malaysian guidelines for the application of CTR and CTX edition for the purpose of applying for this particular license, CTR and CTX. Malaysian guideline for GCP, in case you want to see in terms of how do we conduct inspection and what are the good clinical practice requirement in order to conduct clinical trials in Malaysia, including the CGTPs, as well as Malaysian guidelines for safety reporting of investigational product. So this is whereby you need to report periodically all your SAEs or your SUSAs. So mainly when you're dealing with NPRAs, when you're dealing clinical trials, you need to submit to a SUSA, suspected, unsuspected, serious evaluation. So all these three guidelines are the pillars of our evaluation, and this is very transparent and it's very clear, okay, in terms of the requirement itself, and it is downloadable in our website. And then if let's say CGTP is concerned, you need to refer to this 2016 guideline, which is the guidance document and guidelines for registration of cell and gene therapy products in Malaysia. Okay, you can download it in our website as well when it deals with CGTP. I'm sure it has been explained very how do I say it, very well in terms of previous presentation, in terms of classification of CGTPs. So this is in accordance with our guidelines of registration of cell and gene therapy products in Malaysia, January 2016. We have class 1 and we have class 2. For class 1, it is minimally manipulated and it is intended for homologous use only. Its manufacture does not involve combination with another drug or article or device except for water, crystalloids, or sterilizing, preserving, or storage agent. And as well, it does not have a systemic effect and is not dependent upon the metabolic activity of living cells for its primary function, or if it has such an effect, it is intended for autologous use. Okay, so this is class 1. Okay, meaning that you don't have to apply for any registration or CTI or CTX for that matter if you want to conduct clinical trial. For class 2, product that is that is considered as not class 1, all this registration, CTI and CTX application is required. Okay, when you come to MPRA, if you are a bit concerned in terms of what classes your product belong to, you can always request for classification. We have a specific classification section that one of their core job is to classify which classes it falls on, whether it's class 1 or class 2. So don't be afraid if let's say you can meet us, we can facilitate you. And if let's say you have certain inquiries, you are a bit of concern in terms of what class your products, in your case your CGTP product falls into, you can always ask us to classify it for you. Okay, so we will determine it according to your documentation that is given. So please, in terms of the classification process, so I hope that when you go for classification, you need to give us all your documentation pertaining to your product to help us to classify the product, whether it is under us or other for other department or to be evaluated. Okay. So so in terms of regulatory approval for clinical trials involving CGTPs, as you all know, starting prior to 1st January 2021, it is still voluntary submission. However, comes 1st January 2021 onwards, it is mandatory. So these are particularly the procedure that you need to apply to. Okay, so please bear in mind that for every CGTP research, we have basically two governing bodies. Number one is us, okay, under control of drugs and cosmetic regulation 1984, pertaining to the CTI and CTX application. Okay, if you want to conduct clinical trials in Malaysia. And then the other one is ethical approval. It must go to the main bodies, which is MRC, and MRC will request inserts. Okay, as you can see from Prof. Jamal presentation, to look upon your research in terms of its protocol, in terms of your ethics, yeah, in terms of your feasibility of your studies for their deliberation. For us, it is all related to the quality, efficacy, and safety of the product. In my case, it's quality, safety, and preliminary efficacy of your clinical trials, of your investigational product, for that matter. Right, so this will be the application for CTI and CTX under our CDCR 1984. Clinical trial import license. If let's say you want to import product for the use of clinical trials, or specifically if you want to import unregistered product for the use of clinical trial. For clinical trial exemption, it's to manufacture locally product for the use of clinical trial. So product in this case, in your case specifically, is CGTPs. Yeah, if you have mesenchymal stem cell for that matter, you can. This belongs in our category of product. Okay. So who can apply for the CTI STX? Any PI can apply, or an authorized person from a locally registered pharmaceutical company, which is a sponsor, or a clinical research organization with a permanent address in Malaysia. Okay, so it needs to have a local address in Malaysia. So these are the forms that you need to fill out, and the fees. Yeah, for the CTRL, it's 500 ringgit for each product, including placebo and including the strength. However, for the CTX manufacture, it's still free currently. Okay, this is to boost our economy and boost our capability, a local manufacturer, to manufacture product for the use of clinical trials. So that's why it is free. Okay, it needs a type A license for poisons. It needs a National Medical Research Register registration number. Okay, it needs to be registered there in our registry, in our Ministry of Health registry, prior to applying for this CTR and CTX. It needs to have also, this one is very important. The license permit issuance must be approved by, it needs two approval, must be approved by our part in terms of its IP, and also must be approved by the respective ethics committee. So in your particular case, must be approved by the NSERTs, yeah, and then through NSERTs, the ethics committee, okay, before we can issue a CTIL and CTX. So these are the documents that need to be submitted according to our guideline. You need to submit to us the protocol, the pharmaceutical data, and the investigator's brochure. Okay. So in terms of your CGTP, okay, your CGTP product, you need to cross-reference your guidance, your documents with this following guidance. Yeah, the guidance and guideline for registration of cell and gene therapy product in Malaysia, because we are following that. Okay, you need to cross-reference in terms of all your documentation itself with these following guidelines. So the holder of the CTR and CTX need not necessarily conduct the clinical trial, okay? And the PI or sponsor is allowed to submit parallel application to the MPRA as well as to NSERT. through the respective ethics committee. Okay, for this. So this is to lessen the timeline of approvals. And then there's a caveat here. A CTR and STX will only be issued when both approvals from MPRA and ethics, respective ethics committee are obtained. So this is very important. Now, so this is the non-clinical aspect for the CGTPs. Okay, so what are the requirements that we need for this documentation? For the general aspect of it, of course, we need all these three pillars, which is the individualized non-clinical testing program, the objective of all the non-clinical studies, as well as the goals of these studies. Okay, so in terms of the all these requirements, okay, don't imply that. Please do not imply that we are being so rigid in terms of the requirement. All of these, yeah, rational, absence of study, especially absence of study, you can justify it. If let's say you have a very concrete evidence that shows all some of these requirements cannot be met, you always can justify with us and we will deliberate it further. Okay, not to say that you must have this so-called information, you must have it. Not necessarily. It can be justified if it is not available. Of course, justification needs to be rational. You must have rational justification. You must submit all your what you call this, your journals, all your previous studies that you have conducted, and we will deliberate it further. Okay. The objective of the non-clinical study is very important, like Prof. Rajamal said previously. The proof of concept must be there. The pharmacological and toxicology effect, when you want to predict it for human condition or human effect, must be there. It needs to be justified and it must have its rationale. Okay, and then what are the goals of these studies? Yeah, the goals of all these studies, of course, we need to have a safe dose. Okay, you need to have a selection of safe and efficacious dose. We need to know the rationale of your route of administration, and the treatment must have its rationale in terms of, you know, when you introduce the drug to human, in terms of its duration of exposure. Is it too toxic, or is it okay? Is it stable in terms of its in terms of its potency? And then you really need to identify the target organs for toxicity, and respective parameters when you want to monitor the patients when receiving these therapies. So this is all the concerns, yeah, that we look upon in terms of the documentation provided by the applicant. Okay. Okay, now we go for the clinical aspect of CGTP. Of course, the clinical trial must be designed in accordance to the existing guidelines. And then the trial design consideration, we also look at the trial design consideration. When we look upon the trial design consideration, for us, MPRA jurisdiction, it always related to the investigational medicinal product. In your case, it's CGTP. We did not go for having a full scientific protocol evaluation. We did not go whether or not the blood that has been taken in this trial is okay or not. We only focus in terms of IP related, which are related to the design. Okay, for this example, the evaluation of safety, yeah, that might require observation of subject for a period of time to understand the safety profile. It's all related to the IP. When we look upon the protocol, it's all related to the IP. In your case, it's CGTP. Okay, and the last part is very important: the potential to elicit an immune response in the recipient body in terms of its immunogenicity. Okay, this is how we evaluate the respective investigational product when it refers to CGTP. Okay, so you might wonder in terms of our new requirement, how many CGTPs that we have approved so far. Okay, for now, fortunately, we have one which has been approved recently. This is a cytomesenchymal stem cell, 25 million cells in 1.8 milliliter of cryoprotectants, serums, and 10% dimethyl sulfide in the 2 ml polyethylene carrier. So basically, this is a message can say that we have proof. We have approved so far in terms of its CTIL. So fortunately, it has been approved, and then via clinical trial exemption, meaning that to manufacture locally for the purpose of clinical trial, and this application will be a precedent cases in terms of all the requirements in terms of when we want to evaluate the next clinical trial for for approval of CTIL or even CTX. So this study has been conducted in Hospital Ampang, Sunway Medical Center, as well as Hospital Chancellor Tuanku Mukhris in UKM. This is available. This information is also a public domain. Okay, is also available in our website. So fortunately, we have one to be used as a precedent cases. So hopefully, we hope that we can receive more of your CGTP products to be evaluated through our CTIL and CTX, so that we have more experience in terms of documentation, how we evaluate, and what are the justification that we can allow in terms of these CGTP's product. Okay, so that's it for my presentation. I look forward to your to the panel discussion later to receive all your questions. Thank you.
Thank you for that very clear presentation of the framework for CGTPs. And I'm sure that already many of the questions from the participants have already been covered by your talk. So that's fantastic. Thank you very much. We'll now move on to the next talk, which is on the registration of new stem cell therapy in Malaysia. And for that, we have the privilege of our next speaker, Dr. Evelyn Low, who's the Senior Principal Assistant Director of MPRA, currently in the Biologics section. She received Master of Pharmacy training from the University of Strathclyde and has been working with MPRA evaluating various categories of products since 2008. Her PhD is on biomaterials and cell therapy, and she joined the Biologics division in MPRA after completing her PhD. And she's currently involved in evaluation of biologics, which includes cell and gene therapy products for pre-marketing authorization and assessment of variation and additional indications for registered products. So she has experience that is extremely pertinent to this topic, which is the registration of cell and gene therapy products in Malaysia. Dr. Evelyn Low.
Okay, thank you, Prof. Louis, for the introduction. Good morning. So the last topic for today is the registration of cell and gene therapy products, in short, CGTPs in Malaysia, and this includes stem cell products. So this is the same slide from Dr. Zario's presentation. Just now you have heard about clinical trials. So now I'll be focusing on product registration, a step after clinical trials. As researchers, of course, we hope to see our products registered and used in the market one day. But this talk may or may not be relevant for you right now. Anyway, it's good for you to know for future planning. Regulatory frameworks for CGTPs. So for biologics, the definition is a product whose active substance is made by or derived from a living organism, whether it's plant, human, animal, or microorganism, and may be produced by biotechnology methods and other cutting-edge technologies. So biologics include a wide range of products, such as vaccines, blood products, monoclonal antibodies, recombinant proteins, for example, insulins, erythropoietin, and the newly added one is the cell and gene therapy products. So this is a brief chronology of the framework of CGTP. In August 2012, TWG was formed, and the members consist of various government agencies, researchers, academic and treatment institutions, pharmaceutical industry, manufacturers, and importers. And they came up with the guidance document and guidelines for registration of cell and gene therapy products in Malaysia, which was published in January 2016. In May 2017, a directive was issued regarding the use of this guideline. And in this same directive, mandatory registration and enforcement was postponed to January 2021, which is the beginning of this year, to give the industry time to meet the regulatory requirements set out in these guidelines. So during the period between May 2017 to January 2021, it is a voluntary phase where the industry can voluntarily submit their product for registration. However, up to mid-2020, there was no CGTPs received for product registration. So we actually conducted a survey to understand why this was so. And in December 2020, another directive was issued regarding the implementation of product registration and enforcement in stages. This one I will explain a little bit later at the end of this presentation. And also to exclude HSCT, which is the hemopoietic stem cell therapy. Those that are listed in the national guidelines for HSCT under standard therapy are excluded from the scope of CGTP controlled by the Drug Control Authority. And in January 2021, the regulation of CGTPs began. So this is the same slide shared by Prof. Togo Kamaro just now. Due to the cross-boundary nature of CGTPs, a multidisciplinary approach is required. Therefore, different authorities within the Ministry of Health regulates the different aspects of CGTPs. This is the guidance document and guidelines for the registration of CGTPs in Malaysia. A lot of the information that I'm going to present or have already been presented can actually be found in these guidelines. So what is included inside the scope of this guideline as CGTP products? So is human stem cells, which is the topic of interest of today, human tissue therapy products, human cellular therapy products, genetically modified cellular products, cancer vaccines and immunotherapies, dendritic cell, lymphocyte-based therapies, cancer-based therapies. What is not included are fresh viable human organ, fresh viable human hematopoietic stem cells for direct donor to post-transplantation, fresh blood and blood components such as fresh frozen plasma, unprocessed reproductive tissue such as sperm and eggs, secreted or extracted human proteins such as milk or collagen, sample of human cells or tissue for in vitro diagnostic device, and the newly added one is HSCT for standard treatment as listed in the national guidelines for HSCT. So for those products that are not included under these CGPP guidelines, you can refer to other guidelines published by MOH as listed here. Okay, so the regulation of CGPPs is according to a risk-based approach. So cell therapy products are divided into class 1 and 2. We have heard it covered by Prof. Rahman and Dr. Zaril just now. So for class 1, which is the low-risk products, they have to fulfill all 4 of these criteria: minimal manipulation, homologous use, no combination with drug or device, and no systemic effect, and is not dependent upon metabolic activities of living cells for its primary function. So if it cannot fulfill any one of these criteria, then it would be a class 2 product, which is a high-risk product. And as Dr. Zarek already explained just now, a lot of times it is difficult to classify it. It is not a very straightforward thing. So if you are in doubt, NPRA has a classification, H classification service provided. So because the low risk products, the class 1 CPPs are low risk, therefore it does not need to be registered. It is regulated by site licensure and listing under the Medical Practice Division, donor screening and testing, good tissue practice, labeling, and for post-marketing surveillance, it is subjected to adverse events reporting and inspection and enforcement. For higher risk products, because gene therapy are high risk products, so it will automatically fall into this category. So for class 2 cell therapy products and gene therapy products, they are regulated as biologic products and must be registered with NPRA. So they are subjected to GMP licensing, good tissue practice, registration requirements, which I will go through later, and post-marketing activities such as active surveillance, ADR reporting, and safety updates. And also, they have to keep a patient registry. Registration of CGPPs is according to ASEAN Common Technical Dossier, in short ACTD. So it consists of 4 parts. Part 1 is the administrative part. So this part, you will have your labels, your package insert. Part 2 is the quality part. So at this part, there will be characterization of the cells, the manufacturing process validation, your quality control, means like your testing, your specifications, and also the stability of the product. Part 3 is the non-clinical part. So this includes in vitro and in vivo testing. So this is the proof of concept and also early safety assessment in animals. And part 4 is the clinical part, where trials are done for on humans. So these are like dose ranging studies, clinical efficacy and safety, and also long-term follow-up to assess the long-term effect of the CGPPs. Okay, so our registration, product registration, is done through an online system known as Quest3+. So in this system, you will have to upload all your files according to the different parts, part 1 to part 4. The normal processing time is 245 working days. This is excluding the applicant's time to respond to us. So once your dossier is accepted, then we will review it, and also we will send for expert opinion. And after it goes through a series of meetings, finally, it will be approved with MAL number if everything is satisfactory. There is also a shorter review pathway known as priority review, which is 120 working days, and these are for products for unmet medical needs or during pandemics, like all the COVID-19 vaccines and so on. So just now I was telling you about the survey that we have done. So from this survey, we find out that the industry finds that the part 3, the non-clinical part, and the part 4, the clinical part of the dossier is the most difficult to comply with. The industry has started taking steps to comply with the regulation, but looks like they need a little bit more time. But on the other hand, mandatory enforcement should start without any delay, as little is known about the CGTPs used in the Malaysian market, whether it is safe and effective. Therefore, during this transition period, we have this mechanism, which is called the mechanism for registration and enforcement in stages. So starting 1st of January 2021, all CGTPs class 2 needs to be registered. So this mechanism is only applicable for CGTPs that has been in the Malaysian market for at least 1 year prior to 1st January 2021. So if the product does not qualify for this pathway, then you have to register your product with a full dossier, get an ML number before you can market it. Why at least 1 year was selected is because there are some usage records showing safety of the product and to avoid disruption of patients' access to treatment. So if the product can qualify for this pathway, they have 2 stages of screening. So screening stage 1, we will look at literature review, like products that are similar products that are done by other researchers with the same indication, route of administration, dosage, not their own product. We will take into consideration for screening stage 1. Also, because this product has been used in the Malaysian market already, so the company should also have some sort of their own product at the very least safety. And then we also want to know the company's plan, like their non-clinical trial plan, the clinical trial plan. So all these will be looked at stage 1 screening. So if the stage 1 screening is approved, the CGTPs can be marketed until 1st of July 2022, even though they do not have a complete dossier yet, and even though there's no ML number. For stage 2 screening, then we will request for more. We will need like GMP evidence, we will need release specifications and certificate analysis. The non-clinical studies have to be completed, and at least the early phase clinical trials are on the way. So if this stage 2 screening is approved, then the CGTPs can be marketed until 31st of December 2024. So our aim, our direction that we're moving to is that by 1st of January 2025, all CGTPs to be used in the market has to have an ML number, has to be registered. And most of the stage 1 screening that we have received so far are actually MSCs for many different types of indications. So I want to end with these references, which will be helpful for you. And with that, I thank you for your attention.
Thank you, Dr. Ibrahim, for that very clear presentation. And I'm sure there are many people who are interested. I can see from the questions that are coming with regards to the registration and of these products as well. So we've come to the end of the presentations. Although we have overshot time by a bit, I'm sure you agree with me that these are extremely useful talks and shows the huge expertise they are behind the delivery of these talks. And from the questions that are coming, I can see that there is a lot of interest in these topics. We've come to the panel discussion. And before I start off with posing the questions, I'd like to introduce one of the panelists. All the four speakers are panelists, but we've got an additional panelist with us who has patiently sat through all the talks as well, and he comes with a different set of very important qualities and experience as well. And that is Dr. Muhammad Frodas Abdul Aziz from the Faculty of Law, University of Malaya. Dr. Firdaus holds a BSc in Genetics and Molecular Biology from the University of Malaya and an MA in the Biotechnological Law and Ethics from the University of Sheffield and a DPhil in Public Health from the University of Oxford. He has working experience that's very wide from MOSTI, from the Center of Health, Law and Emerging Technologies at the University of Oxford and the Center of Law and Ethics in Science and Technology in UM. He has also been a trainer for UNESCO bioethics courses. He is currently a senior lecturer at the Faculty of Law in the University of Malaya and is a course coordinator for the Master of Health Research Ethics at the Faculty of Medicine, UM. His research interests are in the legal and ethical issues of bioscience and emerging technologies and the governance of biobanking and stem cell law and policy. So you can see that he comes with an important set of expertise and a different perspective, which I'm sure is extremely important as we now deliberate the topics that have been covered in this morning's seminar. So I also welcome Prof. Datuk Raman Jamal, who has just joined us. I think he joined us earlier. And thank you very much for the talk that he has sent ahead of time. So although we have taken a bit of time from the talks, I don't think it matters. If we are prepared to stay on for an additional 15 minutes, I think we can cover quite a lot of the questions that have been posed to us. I think I'll just start off with one round of questions first before we pick the questions from the panel. And this round of questions that I'm putting around are still preliminary questions that have been submitted by registrants earlier. So we give them sort of first choice. And I would like to ask the panelists perhaps to sort of limit your answer to about 3 minutes. Don't take too long so that we can try to cover as many questions as possible. So I start off with Prof. Tengku Kamarong. You've given us a very broad overview of what is the landscape of research and also given us some idea of what Malaysians have been doing. But what do you think are the biggest challenges facing stem cell researchers in Malaysia today?
Biggest challenge, if you ask me right now, it's actually money. Well, comparing us to all the other countries in terms of research and all that, I think money will be a locked in. It's not just the money; it's also the environment. We don't have enough support for this. So I'm part of the Harvard School for Business, talking about how you talk about business being that land and everything. So for them, right, if they talk about certain technologies for drugs, then they go to certain things. If talking about electronic stuff, they'll go to Silicon Valley. And over there, they have the environment where they have people who are ready to invest, VCs who can see that they can do this. They have entrepreneurs who can actually put your stuff into the market to get it raised into certain funds. And of course, then there's also the government putting a certain amount of funding to help these people to go and start off their business so that they can then go into market to actually buy bonds. Sometimes end up becoming junk bonds, but they still become bonds. So it's not just the it's not just the money, but actually the whole market, the whole environment. I'm sure you heard about that lady who came out from Stanford, ran a business, then suddenly come out that small, one drop of blood to come up with all kinds of tests. I forgot what's the name. And she actually raised, we were able to go and raise people up to about 1.5 billion in terms of funds and everything like that, only to find out that it was not correct. But I'm saying that, I'm not saying that we should follow that step. Of course, that's illegal. But yes, it's the environment. So for me, the challenge is actually the environment. So we're just one part of the environment here in ethics committee in trying to go and regulate. But the bigger environment in terms of the support and all that is the one that's the real problem. It's not the ethics or the regulatory that's the issue. It's actually the whole environment. So I asked him, I remember the professor who said I've already had about 5 spin-off companies and all that. So I said, okay, you said that one of the things prerequisite is location. We don't have the environment here and the wrong location. Why not you move yourself into that location? I said, okay, I've already moved up north, so I have to go across the Pacific Ocean to get my stuff in South as well. So that's the biggest challenge, as I can see. Thank you.
Thank you. I think that's a very important perspective that I put in, which is a food for thought for many of us who want to pursue this new frontier here. I'll just move on to Prof. Raman Jamal. You have covered very well in your talk regarding the different requirements for applications to NCERT as well as to IRBs, and what are the different considerations that have to be taken into account with regards to approvals and reviews of stem cell research applications. But what do you think are the biggest shortcomings in the applications? In other words, what are the researchers falling short of when they submit these applications, which then cause a lot of hold up and going to and fro to get things moving? And to add to that, what would be your advice on how to improve these applications?
Well, thank you, Prof. Dato' Lui, and to all the panel members. Good to see everyone. Well, I had this kind of addressed these questions before, and I believe to move forward because we have had very few proposals being approved, not because we are too stringent. I think Tengku Kamarul Prof. Tengku Kamarul is also in the committee, but I believe because of lack of preparation and also gaps in terms of their protocols and dossiers. And taking the COVID-19 vaccine development as a benchmark in Europe, in US, and so on, you can see the regulators coming in to work hand in hand early on. Maybe this is the way to move forward in terms of improving the successful application of or by the companies or researchers, whether they're based in private or in the public institutions. But this needs a change in paradigm in both our committees and PRAs and so on at NPRA level. Otherwise, we'll get stuck with a proposal getting sent out back because there are so many gaps. I highlighted some examples in my talk. I'm not sure whether a consultancy company or advisory company paid for or pay for service would work, but there would be a conflict of interest there. I believe we should change this approach if we want to make sure Malaysia can move forward. Just like in Australia, Australia is the largest producers of research publication in stem cell research. 25% of publications come from Australia. And they have a very nice sort of framework. 4 companies are approved by the government and given, and 150 million Aussie dollars are allocated for the translational research, including clinical trials. We don't see that kind of sort of framework. Our venture capital companies, as Prof. Tengku Kamarul said, are very risk averse. But back to the question on how to help, researchers and scientists and clinical trialists in this area, I think we should look at a different paradigm in terms of assisting them That's my view there, Prof. I think the atmosphere is right now, I think, because of the COVID-19 vaccine framework, where they accelerated the development to just about a year because the regulatory side came in early, so and guided them through. So this is a good model, I think, that we could see how we could adapt and emulate. Thank you, Prof. Louis.
Thank you. I think that's very encouraging in a sense. I mean, I've been on NSET before a few years earlier, and I also did notice that, it seems like there are so few that can actually make the mark. But there's so much interest in this area and a rich area to be tapped. So I think this is an area that would be good to be able to follow up and help researchers make the mark for tapping stem cell research. Can I move on to Dr. Zariel? You have been involved in regulation and you have given a very clear talk regarding the regulatory framework. But I'm sure just like Prof. Jamal's role here in the ethics approval and regulation, there are also challenges in enforcing the regulation, isn't it, of stem cell therapy in Malaysia. What do you see are the biggest challenges there? Where again are the Malaysian players falling back? How compliant are they, or is it that they're not very aware of what's going on?
Okay, thank you for the questions. I think one of the biggest challenges with, not only MPRA, but with other regulatory authorities around the world is the documentation. How complete is your documentation, and how justified all your rationale in terms of applying for the product to be registered, or even for that matter for conducting CTR and CTX. As you can see from my data that we have so far, we have only one, and that is a very precedent cases whereby they have actually take a leap in terms of having an appropriate documentation to be given to us. And I can see that up to the point of application, they always come to us at the later stage, meaning that when they have, received grants, and then they have been rejected by that particular committee, and then they come to us to deliberate all their documentation. And these have, when you come to that particular stage, you are being pressured by time, and you have been pressured by by grant, closing date, and so on. This is where the challenges is. When we ask for something, it always will be based on international guidelines, and sometimes that requirement are hard to complete in a short period of time. So basically, that's it. Please don't come to us at a later stage. Come to us as what. There's one company. I will talk about other part of classification. This is not CGTP. This is a traditional company who want to promote their product for therapeutic claim. Okay, they come to us at a very early stage. I said to you, how are you willing to invest for international market? If you're willing to invest for international market, you need to spend. You need to invest, to spend and to invest in the requirement that are required for international market. So please, again, we also provide consultation. It's free of charge anyway. Free of charge. You can come in anytime, discuss with us what are your plan, what are the requirements. We will explain to you in every detail. Okay. What are your plan? What are your requirements? What are the requirements that you need to apply in my particular case, CTR and CTX? However, the take-home message is, please do not come at the later stage. Come to us at the early stage so that we can plan. Thank you.
Thank you. I think that's a very practical answer, and I guess it does resonate with many of us who are involved in evaluation in the IRBs and in NSET as well as on the MPRS side. We really do want to help researchers, but honestly, sometimes there's not enough preparation, not enough documentation, not enough awareness of all the various aspects that have to be considered. And they expect that a evaluation can be done just in a few hours, just checking through documents and so on. So I think that's a very practical answer you've given. I'm very grateful for that. So for Dr. Evelyn Low, I think in a sense it's this, you will notice that my question is on the same line. I'm trying to draw out what are the issues that researchers perhaps may not be aware of with regards to all these ethical and regulatory requirements. So for Dr. Evelyn Low, from the registration point of view, what do you think then are the biggest shortcomings you have come across for people who apply for registration? And again, what would be your advice on how to improve these applications? I'm sure we could have a lot more, but I'm sure many of them are not able to make it because of certain shortcomings.
So far, registration specific for CGTP products, like we don't have. But then I can share on like the stage 1 like screening that is like really minimal requirement. A lot like are being rejected the first time around, but after like we give our comments and ask to improve, they come back and then eventually after maybe one round or few rounds, then it is like being approved now. Yeah, so maybe sometimes it's quite difficult to like understand our requirement. Like maybe we will say it like in a sentence or what, but there are many more things behind it. So if, it's good that if it's not clear, then you can come and ask us for clarification. Yeah, and also actually NPRA has a like pre-submission meeting. It's called PSM. This is like a way that you can get regulatory advice related to quality, safety, and efficacy for during development of CGTPs. So if you need it, then you can actually request for a pre-submission meeting with NPRA.
Right. Thank you very much. I think it's good to remind the researchers as well as the participants who are here that we're all out here to help actually, and that there are all kinds of services being devised to help them understand the regulatory requirements so that the success rate could be better. So I'd like to pose this question to Dr. Firdaus, who's been listening very patiently. And you've been listening to all these talks, and you've seen how the framework is going round now for researchers as well as in the sense the importance to public as well as to reviewers and regulators. How well do you think our Malaysian public actually appreciate all these ethical implications of stem cell research and therapy, as well as the regulatory requirements? You know, can we improve this understanding? Where do you think our Malaysian public's appreciation of this lie?
Prof, I think to answer that question, before we talk about public, I think I would like to talk based on empirical research that I myself have conducted and a few other colleagues who have conducted. I think we should focus on how researchers themselves view the current regulation, whether they think that it is clear enough or not in order to facilitate them to, to develop further in this area. Based on the empirical research we have done, we found out that they argued that there's a lack of clarity in terms of the regulation, and let alone the public members who are not really, most of the time, not really well versed in terms of the of this regulation. This can be spotted by the fact that there are many unproven therapies being offered, and there are people who are attracted to these, and they believe that these therapies are genuine therapies. As mentioned by Prof. Tengku Kamarul just now, companies use celebrities to attract the public. So that tells us how much the public is the kind of awareness that the public have. And I also would like to pick on what Prof. Tengku has mentioned earlier, the enforcement. So it's great we have these guidelines. We have many guidelines available, which is great. And we have, NPRA has come up with the revised regulations to include cells, gene therapy as well in the regulation, which is great. But then I always wonder, whether the current regulatory framework is clear enough, and who actually is the regulatory body. We know on paper it's Minister of Health, but is the current, I think we need to seriously discuss this question, whether we need to think of a different framework to have a better regulatory framework, improved regulatory framework in Malaysia, in order to facilitate development, which is what we are, we believe in, and also to protect the public at large.
Thank you. Thank you very much, I think, for giving us that perspective. So now I'm going to try and pick up some questions from the participants that have been posed, some upon registration with this seminar, and some have been in the chat function of the seminar, and some in the Q&A. And I hope to, I'm not sure that we can answer all of them. I think those in the Q&A, the panelists can also see them, but in the chat, I don't know whether they can see them, perhaps they can too. But I'll try to pick out a few from the very beginning. So like to Prof. Tengku Kamarul, there have been a specific question. Is there any sort of research in regards to stem cell for Alzheimer's disease? Is this a focus for Malaysia? Are there any challenges there?
No, I actually answered that in the chat as well.
I see. Okay, sorry.
But no worries. Prof, to be honest, I've looked into the literature, and I'm looking at the different groups that are working on this. I'm not aware of anybody doing works on this, even for preclinical models, as far as I know. Maybe in the lab base, I'm not really sure. But specifically to preclinical models, I'm not really seeing that. And you ask me again, the challenge again is actually about money. To do such research requires huge amount of funding. And since the country is not putting that as a priority, even for the funding bodies, Dato' Rahman Jamal, always get to see him either in Mosque or Mohi and whatnot. Okay, and I'm sure there are Malaysia has other areas that they want to develop further as well. So definitely, I'm not aware that IPSC will be the main area, especially in Alzheimer's. No, thank you.
Thank you. For Prof. Rahman, I noticed that there have been quite a lot of questions which show that there's some not very clear understanding of what is the process whereby researchers get NSET review or approval, if that's the correct word, because I believe that perhaps it's not the right word. The approval still has to come from IRB. But there have been questions like, do studies from MOH and non-MOH sites go to the same NSET body? They think perhaps that they're two different bodies. For non-MOH studies, should the NSET documentation be submitted to the IRB, or should it go to the NSET itself, or does it go to the Ministry of Health, MREC? Would you like to perhaps give some sort of quick direction there?
Yes, I can. I think for MOH centers is very clear because they go to MRAC and MRAC will pass to us after they've reviewed the checklist and all that. For the private centers, I think for those who have their own IRBs and IEC, again, sometimes they forward to MREC, sometimes they forward straight to our committee. So there have been sort of cases like that as well. For the universities, yes, they will be screened at their IRB or IEC level, and then they will pass on to our national committee. This flowchart is available on the MOH website, and that has been sort of in practice for many years. And in fact, we had a briefing session with all the 13 ethics committee plus the ones from universities with the chairpersons themselves or their representatives on this. But if there is a need to revisit this, yes, we can certainly bring this kind of engagement again. I don't mind doing this with our team, especially because chairmen sometimes get changed and so on. So that's my short answer, Prof. Dr. Lui.
Yes, can I perhaps since we are on that topic, the application to NSET is not an online application, right?
Not an online. So usually they
So
send
they
a
send
soft
you
copy to our secretariat at the Bagian Perkembangan under Dr. Jafanita. So who then sort of convenes for a meeting for us within, most of the time we convene within 2 weeks of the acceptance by the secretariat.
That's really good to know. Thank you. So for Dr. Zariel, I picked up a question from the Q&A, and that's, I think, one relevant question. Can the CTIL be transferred from one company to another company?
Thanks for the question, Prof. Lui. Yes, it can be transferred to one company to another company with the route of applying for variation of change of CTI holder.
Right, that's
Yes.
good. Yeah, thank you
Free
very
of
much.
charge anyway.
Very good and direct answer. Okay, for Dr. Evan Low, I think there are also some questions. I don't know whether it's really more to you or to Dr. Zariel, but I think I'll pose it to you first. There have been some questions like, are secretomes and exosomes considered CGTPs?
There are a few products that were sent for classification, condition medium and secretomes, and they were classed as biologics instead of CGTPs because it is a byproduct of the cells, and the product does not contain any cells per se. So they fall under biologics, but the requirements for biologics are still the same. You still have to fulfill part 1 to part 4 of the dossier.
Right, and then to follow up, because it's along the same tone, would cell biologics used for cosmetics and aesthetic purposes be regulated?
It really depends on like the route of administration. Like I know cosmetic, it has to be like topical, so if it's invasive, then most likely it would, like cosmetic wouldn't take it up. Yeah, so again, a lot of things are quite difficult to answer straightforward because we need more information. So it's better if like you submit for classification.
Right, thank you very much. I think there's one question that is posed by an oncologist that caught my eye, and maybe this is going to be a bit heated. And this regarding a few clinics in Bangsa offering class 2 cell therapy. Are these regulated? Maybe I can open this to comments from everybody, from all members of the panel.
Sorry, Prof. Lee, what was that again? Can you just repeat that?
I think there's a question that's posed by from an oncologist in our participants list, and he says there are a few clinics in Bangsa who are offering class 2 cell therapy. So are these regulated? What are your comments on this? And what do you see as issues arising from this?
I've actually written that also that I've typed inside the chat. And for me, it just needs regulated anyway. If what Dr. Zairul has presented and so and the FBI said that such things should be regulated, but I do understand there is a cooling off period right now for this year. If I'm not mistaken, this is what they call it as voluntary registration, isn't it? Right, this point in time. So they will have to take action after that. But I suppose if anybody has concerns, they can always complain directly. And I think it's the NPRA or the Bahagian Amalan or whoever it is, the authority has to take action. And just like Firdaus said, the issue is who wants to take the enforcement of this matter, and they have to have a better, clear governance on this. Correct, Firdaus?
Yes, definitely, Prof. Not only that, we also have, I think we've heard about this. We have a clinic in Damansara that offered treatment for Down syndrome, xenotransplantation, which is clearly prohibited in the guidelines, but somehow the clinic able to provide such services. So I think enforcement is definitely one of the key issues we need to look into in this area of technology in Malaysia.
Dr. Zairul, would you like to comment on this?
Yes, Prof. Louis. Okay, thanks, Prof. Louis, for giving me an opportunity to answer this. I think previously, since the clinical trials or research or treatment involving CGTPs, especially those class 2 indication are not controlled, are not so-called. It's not so-called control. At that particular time, there are so-called no man's land there, so everybody can do anything. But fortunately, we are going for the right direction. So as you can see, 2021, starting from January 2021, once we register it, so it comes with a package. Not only it comes with a registration, it comes with the enforcement, and it comes with a periodic inspection as well. So I have a quite a positive view upon this. If let's say if it's already registered and it's already regulated, automatically it comes under us in terms of whether it is clinical trial import license, clinical trial exemption, registration. For us, we have a specific dedicated team, which is GCP inspectorate team, we call it. So anything that touches or that applies for CTR CTX involving CGTP, so stem cell for that matter, it will be automatically becomes our jurisdiction. So we have a way to monitor them, to check in terms of if any wrongdoings, fraud or misconduct in terms of that particular research. For this, I think current transition period, as rightly said by Prof. Tengku Kamarul, I do think that person need to report to us, explaining the situation, and report to us with the very clear issue, and we will actually investigate it, because now it is really a transition period. We need to see at what point that particular site do the treatment. Is it prior to 2021, all right, or is it for that matter it's just or it's currently done? So we need to do further investigation with regards to that. It all depends on your report. If you're willing to do a report, we're willing to take action. Thank you.
Thank you.
But if I may add, Prof. Dr. Louis,
Yes.
I think we discussed this as well at our insert in our hal-hal lain or other matters sometimes. But I believe waiting for a report is not the best way. That's a passive monitoring. Bagan Amalan has the power to do this, but I believe when we discuss with Bagan Amalan, they're probably under-resourced. We know what's happening, but whether we want to take an active surveillance rather than waiting for reports, and this must change to me. And these clinics are very clever. If you go there, even you check their accounts, there won't be any transactions that are recorded by their accounts sort of division, because money is transacted, well covered, because they know the game, because they're not registered, they're offering this, there's a market demand, and they're just supplying the service. To me, MOH itself, there's no Wakil Bagan Amalan here. We discussed this. They are under-resourced to do active surveillance. But I guess to move forward to ensure the public is protected by, people giving this kind of unproven treatment, I think we should rethink about this as well. That's all, Prof. Louis.
Yeah, thank you. I think when it comes to enforcement regulation, we always run into this issue. Even with regards to professional conduct and so on, and MMC, we always have to either act when there's a complaint, or you have a way of surveilling, which is not easy. But it does not mean that we just sit back and do nothing. I think there are lots of people who are aware, and I think maybe this is a good time to reset our thermometers as far as ethics and regulation goes. There are quite a number of other questions that have been posed, but I gather that the panelists are already answering them through the Q&A or the chat. So I don't think I'll pick up those questions. And also, in view of the time, we actually overshot time by 16 minutes already. I probably just have only one that I'll pick up, and whether there's any special element or more stringency when it comes to applying to IRBs as well to insert when we're talking about stem cell research. For example, with regards to the donor requirements and the donor consent for these products that are going to be used. Would perhaps Prof. Jama want to take a shot at this?
Well, I believe in most cases, the dossier or the protocol that comes lacks this, Prof. Datuk Loy. Companies mention they have collected this from several donors and created master cell banks, but the details are not there. Even consent from the donor themselves are not there, and how this consent is taken and what kind of transaction are done. I believe, and of course, donor screening. Many of these documents lacks this. And I must say again, our guidelines that we have created are no different from that of FDA, TGA, even EMA in Europe. To say that we are stringent locally is totally wrong. And to me, it is the onus of the researchers to look at these guidelines, because that very little difference amongst these big countries that are regulating stem cell. And to say that they are ignorant of this, to me, is a no-no. If you are embarking on this particular technology, to me, it is the researchers' responsibilities to ensure every required document, evidence is really sort of attached to their protocol. I think this is where I think we have sort of researchers wants to jump from step A straight to step M, N, O until Z. When, like I said, the documentation on animal studies, again, everyone is quoting other labs, and yet they have never had experience replicating this in their own lab. How do you expect, any ethics committee to look at this when they have no experience studying the disease at the preclinical model? I think this is something we don't want to become a country where we become a conduit for international companies to come because they assume that our our laws and guidelines are less stringent, which has happened. We have many companies that have brought in products from overseas because they themselves cannot get their products approved in their own country. So Tengku is here also. I think during Prof. Louis' time with us also, we've had this before. And NPRA also knows about this. There was one product from Korea that went to NPRA. I was there as well because they couldn't get approval even in Korea for that product. And yet they tried their chance in Malaysia. So I think the image that Malaysia has a less stringent ruling, so they have that perception probably. Yeah.
Yeah, thank you. Actually, we'll be 20 minutes past our time, and we know it's a Friday. So I think I would like to just go one round to maybe take a minute comment from the panelists. Any last words or message that you like to share? And we just go around. Prof. Tengku?
No, thank you very much, Prof. I think it's been a very good discussion. I think the panelists is also good at answering all the answers as much as we can. I do hope that if you have any questions, we can try to help. We really would like to help. I think Dr. Rahman and myself, I'm sure Fidel's, so we all envisage of wanting to see a country quite advanced in the area of stem cell therapy, but it's just we want it to be done in the right way so that others can respect us rather than see us as one of the banana republics that they can try.
Dumping ground.
Thank you.
Prof. Rahman, any more to add?
Nothing. I just want to thank you, Prof. Dr. Loy, for inviting me, and also to Dr. Alex Pang, who's been communicating. I'm sorry I couldn't go live, but I hope the recorded session went well. Thank you.
No problem. Thank you so much, Tato.
The other panelists, can I just go around? Dr. Firdaus?
Yes, I just want to say that I totally agree with what Prof. Rahman mentioned just now, that we need an active surveillance rather than being more passive. And I think what's lacking in Malaysia is advocacy, communication with the public, because you asked about the public, perceptions towards our regulation. I think yes, we have a venue for the public to make complaints to the authority, but I think there's also must be a communication from the regulators to the public as well. And other than that, I'm really hoping to see a better and improved regulatory framework in the future, because we have great scientists in Malaysia, such as Prof. Tunku and also Prof. Rahman and you yourself, and we need that infrastructure to be there to support researchers in Malaysia, definitely. And thank you for inviting me.
Thank you very much for your perspective. Dr. Zaria, any last words?
For me, last word is that thank you for inviting me to this event. And then please, in terms of, if the applicants have any concern and then you need free consultation for us, our door is always open to discuss. And then we need a proper documented application, okay, as a precedent cases. Currently we have just one, we need to have more. So please, if you have any, you need consultation, please contact me and we will discuss further. Thank you.
Thank you so much. Dr. Evelyn, any last message, last words?
I think we look forward to receiving your either stage 1 manual screening or your product registration. Yeah, so if there are any questions, you can always contact us. Thanks.
Thank you very much. I think you can see from this seminar and the panelists that actually everybody is trying to help in this area, but not at the expense of ethics. And I think that's extremely important. And that's why this scrutiny has to be carried out. So in a sense, we're all in it together, perhaps wearing different hats, but we are not against each other. We're only here because we want Malaysia to progress and to harvest the benefits of this stem cell therapy area. But we have to be aware that safety is considered, the public is protected, and there's a proper risk-benefit analysis. And for that, there's no shortcut to it. There has to be the important documents there. There have to be the truth. There has to be evaluation of science behind things before we set out to do anything. So I think that would be my last words. And of course, not truly my last words. My last words is to really thank the speakers and the panelists and the Secretariat of MRRDC for their contribution. I think I have found this a very, very enlightening and useful seminar, and I hope that the participants have found that to be so too. It is just a very preliminary attempt, just a so-called 2 hours, in fact now 2 hours and 25 minutes of discussion, but I hope it's been time well spent. Thank you very much for joining us, and a special thank you for the speakers and the panelists.
Thank you, Prof.
Thank you, Prof. Thank you.
Thank you, Prof.
Thank you.
Thanks, everyone. Thank you. See you.
Thanks, Alex.
And Alex, any last words from you
No, I
to
just
the
want to.
participants?
No. I just want to say thank you so much for your participation and all the panelists and also the speakers. And this is truly enlightening me as you know, one of the members in ethics committee and also as a researcher in stem cell field as well. Thank you so much, everyone. Thank you.
I think everyone who is officially registered will get the e-certificate sent from the Secretariat, right?
Yeah, there will be a post webinar email from us after this. Yeah. Okay. Thank you.
Good.
Thank you so much.
very
Thank you, Prof. Thank you, Dr. Zariel, and thank you,
Have a
Evelyn.
good day.
Thank you so much, everyone.
Bye.
Bye.
Bye.